Hello, this is Ern. This is an online recording for the topic Inheritance 2. These are the learning outcomes of the topic. And this is the contents overview for the entire topic. I have divided the entire topic into 4 different components. Let's begin with the first one, dihybrid crosses involving autosomal linkage. Well, some recall of our prior knowledge. You have learned previously that genes are found on chromosomes. Chromosomes, there are 2 types, autosomes and sex chromosomes. If the genes are found on sex chromosomes, we'll refer to them as sex-linked genes. Of course, we understand that each chromosome can carry more than one gene. And when we are looking at 2 different genes located on the same chromosome, we can refer to them with a term known as linked genes. Look at this diagram as an example. Gene A and gene B will be regarded as linked genes. But if we explore gene A and gene C, we can say that they are not linked genes. So what's the definition for linkage? As mentioned previously, linkage itself is an event or is a phenomenon observed when we are exploring or we are looking at 2 different genes that's found on the same chromosome. When they are found on the same chromosomes, the implication is that the alleles of these 2 genes, they do not assort independently from one another. Rather, they are inherited together as one unit if crossing over does not occur. Because of this, whenever we are dealing with an experiment investigating linked genes, we can expect the results to deviate from those that's expected from Mendel's law of independent assortment. The next term we want to look at is crossing over, which is really a revision of something you have learned previously in meiosis. This is an event that occurs during prophase 1 of meiosis, whereby homologous chromosomes can pair up into bivalents via the formation of chiasmata. During this event, there's an exchange of genetic material between the non-sister chromatids of homologous chromosomes, and there is an actual breaking and rejoining of the chromatids, resulting in recombinant chromosomes being formed. Do note that this crossing over event happens by chance. Right, let's have a little activity here to just illustrate the understanding of crossing over event. You will see that there is a germline cell here with 2 lines representing a pair of homologous chromosomes. On each chromosome, you will find 2 linked genes, gene A and gene B. Can you draw right now into the box? On the left, what happens or what are the gametes formed when there is no crossing over occurring? And in the box to the right, what are the gametes formed if crossing over do occur? Alright, on the box to the left, when there's no crossing over, we will see the following 2 gametes. The capital letter A gene, allele, will combine with the capital letter B allele to be going into the same gamete. On the other gamete is the small letter A and the small letter B. We refer to these combinations, the parental combination, because they follow the exact same combination that you see in the previous germline cell. In the box to the right, if crossing over do occur, you will still see the parental combination as well as 2 new combinations we refer to as recombinant. The reason why you will see the parental combination is because crossing over is a chance event. So sometimes it occurs, sometimes it doesn't. So in the event that it does not occur, we will get back the parental combination, which is actually happening in larger numbers. In this chance event that crossing over do occur, you will form the recombinant combination, and this actually will be observed at a smaller number. Right, so this is another representation of what happens when we have the gamete formation, but this time around for unlinked genes. You will see that they will have an equal chance of forming 4 different types of gametes, 4 different types of gametes that arises due to the idea of independent assortment. If we look at them, gene A and gene B being linked, The idea is that the formation of gametes itself will give rise to two types of parental as well as two types of recombinant. And as discussed, the parental combination will arise in larger numbers, while the recombinant combinations will actually be observed in smaller numbers due to crossing over being a chance event. So how do we represent linked genes in genetic diagram? So when we represent, or rather when we understand that the genes are linked, we will represent them using a line drawn across to represent the chromosome. So the diagram to the left, you will see that it is represented in a horizontal configuration, while the diagram to the right is in a vertical configuration. Both are equally accepted. However, do note, if you look at these two diagrams, while they might have the same phenotype, genotype-wise, they are not the same. This is because the diagram to the left, capital letter A allele and capital letter B allele are found on the same chromosome, while to the diagram on the right, capital letter A allele is linked to small letter B allele. So genotype-wise, these two diagrams are not the same. So what does this genotype mean then? Please note that this is an incorrect representation and is one of the most common errors that students commit. If you look at this diagram, on the same chromosome, you will see capital letter A and small letter a. This is incorrect because both alleles of the same gene cannot be found on the same chromosome. They should be occupying the same gene loci on separate homologous chromosome. Let's look into a case study right now. Please read the question. And what do you observe? In this question, this is a typical test cross of an F1 plant and shows the results of the F2 plants with their genotype and with their phenotype. So this cross itself, let's do some kind of a recalling of prior knowledge. It's a typical test cross of a double heterozygote. The expected cross should give you a ratio of a 1-1-1-1. Let's look back into the observed results. You will notice that the results actually deviate drastically from that of an expected 1:1:1:1 ratio. Next, you will also observe that the initial idea of the parental phenotypes are actually greater proportion than what you expect. Notice the tall plants with red flowers as well as the short plants with white flowers. These two phenotypes which follows the original parental phenotype, they are occurring larger or greater proportion than what you expect, while the recombinant phenotypes of tall plants with white flowers, short plants with red flowers, they are occurring in smaller proportions than what you expect. This kind of deviation will further support the idea that these two genes that encode for flower color and height are found on the same chromosome, i.e. they are linked genes. Once you reach this deduction, the understanding is that the explanation behind it is that when there's no crossing over occurring, you will only have two parental types that's occurring in large numbers, while if there's crossing over occurring, you will have 4 different types of gametes, of which the parental combination occurs in larger numbers, while the recombinant combinations occur in smaller numbers, because crossing over is a chance event. Let's look into the idea of how we're going to represent our understanding now, having deduced that these two are linked genes. How are we going to represent them in a genetic diagram? The genotype wise for a tall and red plant will therefore be drawn with the line representing linked genes with capital T and capital R on the same line. For a short and white plant, it will be a small letter T and a small letter R on the same line. Gametes form wise, we will actually take out one particular chromosome. There will only be one possible combination because this is a double homozygous dominant. While for the short and white plant, being a homozygous recessive plant, there is only one possible combination of the gamete, which is small letter T and small letter R. When you put these two gametes together, you're going to get this possible F1 genotype. Subsequently, there is a test cross of the F1 plant. The F1 test cross will involve firstly the F1 plant, as well as a homozygous recessive plant, which is short and white, small capital T linked to small capital R. The gametes formed, do note that the tall plant, which is heterozygous, will give rise to 4 possible gametes, of which, as discussed previously, 2 combinations are the parental, which will occur in higher numbers, and 2 combinations will be recombinant, which will be occurring in smaller numbers. The homozygous recessive short white plant will give rise to 1 possible gamete. Once you are able to deduce the number and type of gametes that's formed from the various plants, you will then attempt to plot out the Punnett square. The Punnett square itself, you just need to insert the gametes into the corresponding slots and work out the genotype of the F2 plants here. Right, it is advisable that you actually put in the corresponding phenotype to the genotypes in the Punnett square directly. Right, after you have plotted out the Punnett square, we will proceed on to actually tabulate the phenotypes of the F2 as well as the ratio that you will see. So F2 phenotype itself, you have 4 types: the red tall, white short, red short, and the white tall plants. The expected ratio that you will get is 1:1:1:1. However, the observed numbers will deviate, and I'll expect in your genetics diagram to give the explanation why they will deviate. So for instance, these 2 numbers correspond to the parental combination, and they would be observed to be higher than expected, with the explanation that the parental combination of alleles are inherited as 1 unit. The other 2 combinations, we refer to them as the recombinant combination. It is expected that you are able to provide the explanation that the numbers will be lower than expected because the recombinants are formed as a result of crossing over, which is a chance event. Let's look into the next concept with regards to linkage of genes. Understand that when we are looking at 2 different genes that are linked, whenever they are closer to one another, this reduces the probability of being separated by crossing over. That gives rise to actually a lower proportion of recombinant types. Let's look at an example over here. If we look at this pair of homologous chromosomes right now with gene T and gene R at 2 different locations, when they are separated across on the same chromosome, there will be a recombination event if the crossing over event occurs anywhere between these 2 genes. But if the crossing over occurs anywhere outside of this region, namely here, or even or even if it does not occur at all, there will be no recombination at all. So I repeat, if crossing over event occurs anywhere between these 2 genes, you are going to see the recombinant types being formed. However, if crossing over event occurs outside of the event or outside of this region, you will only see parental types being formed. Therefore, I would like to introduce to you the next concept of total or complete linkage, whereby the 2 genes are so close together such that the crossing event is not possible to occur. It is not possible for the crossing over event to occur, whereby therefore only the parental combinations will be generated. Let's look at this example again. Right now, in the example of complete total linkage, I've shifted the 2 genes to be close together to each other. In fact, so close such that there is no possibility of crossing over event occurring. Because of this, you will only expect to see parental combinations observed in the gametes form. Right, so this is the follow-up of the case study 1, but right now with a twist. What about exploring the same case study 1, but in the event whereby the genes are so close together to one another that we refer to them as total linkage. Back to the test cross of F1, recall that in the test cross of F1, the tall and red plant will give rise previously to 4 different gametes. In the event of total linkage, you will now only see 2 possible combinations, the parental combinations. The recombinant combinations will not be observed because in the event of total linkage, there will be no recombinant combinations being formed. When you bring in the idea of these 2 gametes as well as the gamete from the short and white plants to form the F2, you will see just these 2 possible phenotypes in the F2 plants. The ratio expected will be 1:1. Let's go into the first practice question 1. Take some time to read through this question. Alright, and it follows up firstly, part A, to show you a table showing the results of the cross. You are asked to actually come up with the expected numbers if there is no linkage. Alright, so what are the steps in order to find out the expected ratio, rather the expected numbers? Firstly, you have to find out the expected ratio of the cross. Note that this is a test cross between a double heterozygous and a homozygous recessive. The expected ratio of the test cross you observe will be 1:1:1:1. The step 2 involves adding the total number of offsprings together. You will get 2640, divide by 4, and you will get 660 each. For each phenotypic class, you will see 660 offsprings. Let's put the numbers into the box. Next, part B, state the meaning of the term linkage in this context. So in this context is key because you are required to contextualize the definition of linkage into the question. So this will be the idea whereby you have the gene that code for the eye color and the gene that code for the wing types being found on the same chromosome. This will give the definition of the term linkage. Next, you are required to draw a genetic diagram to explain the results of this cross. So they have provided for you the symbols. Now you actually have to proceed on to come up with the genetic diagram. Recall when you are first faced with a genetic diagram, the first step is always to try to deduce the inheritance of the traits. So when you look at the table, what you observed is that there are 2 phenotypic classes that are higher than expected, and there are 2 phenotypic classes that are lower than expected. This seems to flow in line with what you have learned just previously, whereby you have 2 phenotypic classes that's higher than expected, and when you have 2 phenotypic classes that's lower than expected, it seems to point towards the idea of linkage. So the 2 genes are linked in this question. You will have to then deduce which are the parental and recombinant combination. And as learned previously, the 2 phenotypic classes that's higher than expected, they will be your parental combinations. The 2 classes that's lower than expected will be your recombinant combinations. And when you draw the diagram, you will represent them in a linked genes configuration with the line straight across to indicate that these 2 genes are linked. So after that, you form the 4 gametes, of which 2 are the recombinant gametes, 2 of these are the parental gametes. Put them into the Punnett square, just like we have done so in case study 1, and come up with the corresponding phenotype and genotype of the F2 plants, or F2 flies in this case. Together with these 4 particular F2 pheno and genotype, you will see that they will correspond to the numbers as indicated in the observed numbers. Provide the explanation of why you will see 2 phenotypic classes higher than expected and 2 phenotypic classes lower than expected. One of the things that I want to cover now is the idea of how do you actually represent the genotype into the correct symbol for linked genes. Take note, if it's an unlinked genotype, you will represent a double heterozygous in this way: big R, small r, big N, small n. However, if the genotype is linked, you can actually have two different types of representation. The first one whereby the big R and big N are found on the same chromosome. The second possibility whereby your big R and the small n is found on the same chromosome. Do note that these two genotypes, while they are different, do give rise to the same phenotype of red eyes and normal wings. However, in your genetic diagram, you have to be very clear in which of this representation is the correct one. So how do you know whether is it the one on the top or is it the one on the bottom? To do so, we have to go back to the table. Let's go back to practice question 1, the table of results. When you see that they are the parent, when you have deduced through the expected ratios and the observed numbers, which two are the parental phenotypic classes and which two are the recombinant phenotypic classes, from the parental phenotypic classes, you can deduce that the alleles for red and normal are linked. So you will represent them with the big R with the big N on the same line. With the second parental phenotypic class, you will notice that the purple eye as the vestigial and the vestigial wing alleles are linked. So you represent the other chromosome with small letter r and small letter n. So all in all, the gist of this is that you will have to go back to look at the parental combinations to deduce which are the alleles that's originally linked on the same chromosome. With these two representation in line, we can then deduce that the correct representation in this question will be big R linked with big N and small r linked with small n. Right, let's go on to the next concept of recombinant frequency that determines the map distance. Recall that when we have this crossing over event occurring, recombinants are actually able to be formed. Remember this animation that I was showing you earlier on, where we have the same gene, the same chromosome, whereby we have two genes, the gene F and gene L this time round. If there is a crossing over event occurring between these two genes, take note that this will give rise to your recombinant types. But if the crossing over event occurs outside of this region, there will be no parental types. Let's try pulling the two linked genes apart further. What do you notice now? There is actually a larger region at which the crossing over event can potentially occur to give rise to recombinants, while there is a smaller region that when crossing over occurs, there will be no recombinant formed. So in the event where the genes are actually being pulled apart further from each other on the same chromosome, we will notice that there will be an increase in the number of recombinant types being formed. So what does this all imply to us? Is that when we actually have two genes that's closer or further apart, they will influence the number of recombinants that we can observe. Conversely, therefore, we can actually use the proportion and number of recombinants that we observe to determine the distance between the linked genes. To determine the crossover value, what we'll do is we'll carry out a test cross first. So when we do a test cross, this is similar to what we have conducted in practice question 1 and case study 1. The expected ratio will be 1:1:1:1. Using this formula, whereby we do a crossover frequency, is equivalent to the number of recombinants over total number of offsprings multiplied by 100%, we will find out that this will be what is the map unit distance between the two different genes. Note the units used in this case, where 1% of crossover frequency will be representative of 1 map unit of map distance, which is also represented by 1 centimorgan as the unit. Why is the understanding of crossover frequency so important? When we are able to understand the distance between two different genes on the same chromosome, geneticists can actually produce something known as the chromosome maps, that can show you all the relative location of genes that's found on the same chromosome, as well as the distance between them. Let's look into case study 2, and we try to actually factor in recombinant frequency into this question. In this question itself, again we are looking at F1 flies this time round that will test cross with double recessive flies, and they show you the results of the 4 phenotypic classes that were obtained. The 2 phenotypic classes that's larger than expected, and there will be 2 phenotypic classes that's lower than expected. They will be your parental and recombinant combinations respectively. And of course, I think by now you'll be quite familiar with this, the idea that because the observed numbers deviate from your 1:1:1:1 ratio, you can deduce that these 2 genes are linked. When we work out the recombinant frequency, all we have to do is to add up the total number of the recombinants with the total number of offsprings and multiply it by 100%. This will allow us to find out that the recombinant frequency is actually 17%, which is actually going to be translating to 17 map units apart for these 2 genes on the same chromosome. Let's take a short while to explore and do one more revision practice question in the context of case study 1. What do you think is the map distance between the genes in case study 1? Right, if you refer to case study 1, the number of recombinants in total will be 11 plus 14 divided by the total number of offsprings multiplied by 100%. You should get 12.5% crossover frequency. That indicates to you that these 2 genes in case study 1 are 12.5 map units apart on the chromosome. Next, let's carry on to practice question 2, where we actually have to map 3 different genes on the same chromosome. So in a breeding experiment, the crossover value of 2 genes P and Q were found to be 4%. The crossover value of gene P and R was 1%. So now they're asking you to find out, so what is the crossover value of genes Q and R respectively? So in order to do that, it is advisable that you attempt to plot a chromosome map as part of your working. So let's start with the simplest. P and Q were 4% apart, so you will plot it as a 4% as the map distance. They mentioned to you that the crossover value of gene P and gene R was 1%. Because the crossover frequency itself indicates to you the magnitude but not the direction, it is possible for gene R to be located either on this side to the left of P or to the right of P, as indicated in this diagram now. So it could be 1% to the left of P, or it could be 1% to the right of P. With that in mind, therefore the crossover frequency or the crossover value of R to Q can either be 3% or 5%. So therefore the option will be D. Let's have another practice question to see how we can determine the crossover value for another 3 genes. So you are given this information whereby you have all these crossover values between the corresponding genes. Usually in the working itself, what you should do is you will draw a line first to represent a chromosome. Insert the genes with, you are recommended that you will insert the genes with the least crossover value, although really the order itself can be switched around. So let's start. If you start with S and P as the 6% between the 2, you go on to do the next piece of go on to input the next piece of information that you have, R and S, that's 8%. As I've mentioned previously, The crossover value itself indicates the magnitude but not the direction. So if R and S is 8%, it is possible for R to be located to the left or to the right of S by 8%. To actually determine which particular location of R is the most likely position, we can look at the other information to try to eliminate the position of R. So R and P is 14%. So if R is actually the position to the right of S, it is not possible for you to have a 14% crossover value with P. Therefore, I will eliminate this potential R and stick with the one to the left of S. That works out to be exactly 14% from P. What about Q? Because the information given next was P to Q is 24%, it is possible for, likewise as I said, no indication of direction, it is possible for Q to be located 24% to the left of P or 24% to the right of P. So in this case, we will need additional information for us to deduce from here. So all we can do, at least with the information that is provided, is we know that it's either QRSP or RSPQ. The next question, practice question 3. So take some time to consider what is the correct order of genes on the chromosomes for this question. Right, so let's try working together for this question. So firstly, P and R is 5% apart from one another. We can put in the 35% between P and Q, although you can also go by the order from smallest crossover value to the largest. It's alright, but let's try with 35% for P and Q as a distance. Again, there's no indication of direction, so it can be 35% to the left of P or 35% to the right of P. We eliminate that when we are able to see R and Q is actually 40% apart from each other. So therefore, this particular Q position is unlikely to be the answer. So we will actually remove that and stick with the Q to the left of P. So now 35% and 40%, it fits into what the table of information has given us. Let's try putting S into this. It will actually be 10% from Q and 30% from R. So therefore, I will expect S to be somewhere between here. The correct answer will therefore be QSPR. Note that because this is the indication of magnitude but not direction, the answer can also be RPSQ, or the opposite orientation. Finally, let's look at something known as the double crossover event. A double crossover event is whereby you have two crossover events. The first crossing over to give you the recombinant, and a second crossover that actually return the genes back to the original position. When you have a second crossover event, you will lead to a decrease or a lower than expected number of recombinant combination. So how can a question be asked about a second crossover event? Is whereby they provide you with certain information about 3 different genes. For example, in this question, you notice that they gave you 10% crossover value between 2 different genes, 10% between another 2 different genes. It is not possible for the crossover value between the first and the third gene to be 15% when it should have been 20%. Therefore, we are looking at a lower than expected recombinant number. This can only best be explained by the fact that a double crossover event has occurred. Alright, so that is the end for the first topic of inheritance 2. We'll take a break here.